Okay, so it's my great pleasure to introduce today's Pau Hana speaker. Dr. Randall Kosaki is the research coordinator at NOAA's Papahānaumokuākea Marine National Monument. But as Ken alluded to, I know him much more than that. We first actually met when we were teenagers about 40 years ago, which sounds like a long time ago. Well, because it was a long time ago. We were brought together through our shared passion for coral reef fishes, and we've maintained our friendship, and we've been colleagues ever since. He introduced me to my wife of 26 years, and he was the best man at our wedding. And over the years, Randy and I indeed have shared many adventures. We both learned to do deep dives together, mostly through trial and error. We're both still here because somehow we've managed to save each other's lives slightly more often than we've managed to almost kill each other. And yes, I could tell you many stories about him, but I won't. Because he can tell you just as many stories about me. And we sort of have this mutually assured destruction pact that requires non-trivial quantities of alcohol to break before we get into the storytelling mode. But in all seriousness, I've had the great pleasure to join Randy and his team on research cruises to the monument almost every year since 2009. And while my main research focus has been on searching for new species of fishes and things, Randy and his team have been the true pioneers in conducting important quantitative hypothesis-driven research on these deep coral reefs. So I'll shut up now and turn it over to Randy to share all that research with us.
Well, thanks a lot, Rich. Thank you for honoring our pact, because this could get ugly pretty fast, mostly because I have the floor now and you don't. I've been blinking in and out because I just noticed that this has my name down as Reese Kosaki. I am most definitely Randy Kosaki. Reese is my 15-year-old son who often does things to my computer that I don't know how to How'd you do that?
I just changed it.
Oh, okay, great. Yeah, good. I'm near a computer literate, but you know, Reese would have been okay too, because back when I used to have a name, I was Randy Kosaki, but now wherever I go, I'm just Reese's dad. So that would have been fine too. But yeah, Rich, thanks for that introduction. Rich and I have known each other far too long. Rich is almost, as he said, he's almost caused my funeral on several occasions. Yeah, back when we were much younger and much, much stupider, we did a few really interesting dives together, but we're all better now, right, Rich? And so now I'm gonna tell you a few stories from the latter part of our lives when we do it the way you're supposed to do. So if I can figure out how to share my screen. There we go. Okay, is that showing up? Yeah, so what I'm going to talk about today are some of the dives we've done exploring Papahānaumokuākea, or the Northwestern Hawaiian Islands, with Rich and our NOAA team and the Bishop Museum team. But first I'm going to start off with some background, because Papahānaumokuākea, even though it's 2/3 of our state, it's not exactly a household name. We started out life as a reserve designated by President Clinton back in 2000. And so I think, I don't know, can you see my cursor? This inner little blue sausage, these were our boundaries from 2000 until 2016. So we were first created as a monument by President Bush, and in 2006, we weighed in at that time at 139,000 square miles, which already was enormous. But in 2016, President Obama expanded our boundaries out to the limits of the United States Exclusive Economic Zone. And overnight, we quadrupled in size, and we're now 580,000 square miles, which is, yeah, that's kind of hard to get your head around an area of ocean that large. Suffice to say, we're the largest conservation project under US flag, and one of the largest marine protected areas anywhere in the world. We're larger than Texas. And so the coral reefs, which are arguably the crown jewels of the entire monument, are still only about 3% of our real estate. Most of the real estate in the monument is deep ocean, not coral reef. But our reefs are some of the most unique in the United States. We have the only true atolls in the United States up in the Northwestern Hawaiian Islands. Now, of course, we have similar structures in American Samoa and surrounding territories, but technically those are not one of the states. So this is one of the things we can lay claim to as being unique to Papahānaumokuākea. What we're best known for, of course, are our protected species, our threatened green sea turtles and our critically endangered Hawaiian monk seals. As Rich Pyle said, we met many years ago. Rich and I actually, our first jobs right out of college were at the Waikiki Aquarium. And this is just kind of a personal aside. But back in the day when Rich and I were growing up, you rarely saw sea turtles. It really was something to write home about. Whereas now, if you go out to Laniakea on the North Shore or elsewhere, the tourists literally are stepping all over turtles in 1 foot of water. So they've made an amazing numerical comeback since they've been protected. And yet they have a life history stage that is intimately tied to the Northwestern Hawaiian Islands. And that is that 90% of the sea turtles in the Hawaiian archipelago go back to one atoll, to Lalo or French frigate shoals, to nest. And so even though they've made this amazing comeback, they're still tied to a small number of very low-lying sand islands. And these sand islands, as we'll see later, are very much under threat from the triple whammy of climate change, which is seawater acidification that inhibits corals from secreting their skeletons. And of course, it's coral sand that builds these islands. Sea surface temperature rise, which again can cause coral bleaching and death. And then increased frequency and severity of very large storms. And these hurricanes, of course, they're thermal engines. And so they feed off of hot ocean water. And we're making that problem worse for ourselves and for these animals by continually warming the planet with greenhouse gases. But on a personal note, back when Rich and I worked at the aquarium had two just really gigantic green sea turtles in captivity. They had been in captivity for over 20 years, I believe, and they were just too big and ornery to keep in the turtle pond anymore. So we tagged them and rather unceremoniously dumped them over the seawall at Waikiki, figuring we'd never hear from them again. And the following summer, after 20 years in captivity, those two turtles turned up on the beach at French frigate shoals. So there really is this intimate relationship between these turtles and small slivers of nesting habitat in the Northwestern Hawaiian Islands. Now, everybody loves our turtles and seals, but I'm a fish biologist. So for me, the Northwestern Hawaiian Islands is all about the fishes. And this, of course, is an ulua, or giant trevally. If any of you fish, shore fishermen, or if you have relatives or friends that fish, every shore fisherman's dream is to join the 100-pound club. Once in your lifetime to catch a 100-pound ulua. And for most people, at best, it's a once-in-a-lifetime event. And when we're diving in the monument, we see 100-pounders, 150-pounders every day. Literally every day we go in the water. Back when fishing was allowed, one of the crew on one of our NOAA ships actually caught a 165-pounder on a hand line. We weighed it and let it go. But a 165-pound fish is bigger than me. I mean, that is an absolutely enormous fish. And so these are things that you almost never see here in the main Hawaiian Islands. Sites like this where this fish that is very much prized and very heavily hunted, both by pole fishermen and spear fishermen. You just don't see these big packs of ulua here, whereas up in the monument, it's not uncommon to see schools of 30, 40, 50 of these big fishes. And they're completely wild. They have never seen human beings before. They're completely fearless. So this happens once in a while, maybe not every year. I always have to tell our newbie divers, like when we take University of Hawaii students or other folks that haven't dived with these fishes before, I have to caution them, don't swim around with your hands up like this. Keep it tucked in tight, because these are 100-pound plus, completely wild, meat-eating fishes. They have big teeth, and they do things like this. And I honestly don't think this is the result of aggression, although it seems like an aggressive move to bite someone. Because fish don't have hands, and I think of it more as curiosity. If we see something we're curious about, I can pick it up and go, what is this? And well, it's not actually that interesting. I'll put it back down. These big fish don't have hands. So if they're curious about something, like your hand, they'll pick it up with their mouth. When they realize it's not as tasty as it looks, they'll spit it out. Notice these are just superficial scratches. This guy didn't need stitches. He just needed Band-Aids. So they really are biting fairly gingerly. I'm not sure they realize that they probably could very easily have actually removed this diver's hand from his body if they wanted to. But they're not attacking out of aggression. I think it's a love bite driven more by curiosity. But this kind of thing happens up in the monument. And for a fish that's hunted so heavily here in the Hawaiian Islands, this is just, there's something really bizarre about this. It's like man bites dog, you know, fish bites fisherman. This thing just isn't supposed to happen. There's just something fundamentally wrong with this. And then there's the sharks. These are my favorite. You know, these are the so-called apex predators that are at the very top of the food chain. And if any of you dive here in the main Hawaiian Islands, you probably know that you rarely see sharks, maybe a couple times a year if you're lucky. These are some of the most beautiful fishes in the ocean. I could watch them all day. So you rarely see them here, but up in the northwestern Hawaiian Islands where there hasn't been a 1,000-year history of fishing, these sharks, we'll see them in schools of 50, 60, 70 sharks. And this is actually one of my favorite things to do. After we're done with our scuba dives, it's to ditch all that heavy gear, just go in with mast fins and snorkel and skin dive and swim with the fishes. You know, when you're skin diving as opposed to scuba, you're silent, you're much more nimble, you're much faster in the water. And the fish recognize that. I think the greatest compliment that a fish can pay to a diver is to completely ignore him or her, because that means they just look at you as another fish. Now, as you can see, these sharks are following me around, so they're looking at me as maybe not just another fish, but probably just the weirdest fish they've ever seen. But again, skin diving really, I think, is the way to see the northwestern Hawaiian Islands, because your impact on the behavior of the wildlife is then minimized. Unfortunately, a lot of the work we do, though, requires more than that. And we do fish counts, and these fish counts produce data like this. And so these two pie charts kind of tell the story. The pie on the left represents the biomass of fish in the northwestern Hawaiian Islands. And as you can see, these apex predators are over 50% of the biomass of the fish on the reef, are big fishes like sharks and ulua that you really don't see too many of here in the main Hawaiian Islands. Whereas this pie on the right is the same thing. It's a biomass of fishes, but this time in the main Hawaiian Islands. And these apex predators are 3% of the biomass on our reefs here. So really, it's an astounding difference in how many of these fishes you see. And if these pies were really proportionate, the northwestern Hawaiian Islands pie should be 3 times the size of the main Hawaiian Islands pie, because overall fish biomass is 3 times greater than here in the main Hawaiian Islands. And of that greater biomass, 54% of that are these big fishes, ulua, uku, tiger sharks, Galapagos sharks. And so it really is kind of a window back in time. This shows you what a Hawaiian reef probably used to look like 1,000 years, 2,000 years ago. And you know, with a little better management, maybe this gives us a glimpse of what our reefs could look like at some point in the future. So this is what everyone expects when you talk about coral reefs. In a beautiful coral, hundreds, thousands of pretty colorful coral reef fishes. And certainly we have a lot of this in the northwestern Hawaiian Islands. But Rich and I, what we like to do is to access the deeper half of the coral reef habitat. This figure kind of illustrates most divers on scuba, breathing air, your real hard floor, your hard limit is about 200 feet. And most divers really work in the depth ranges of maybe 30 to 70 feet. And this is recreational divers, but this is also scientific diving. So most of the research, most of what we know about coral reefs is based on diving probably to depths of less than 100 feet. And yet, reef building corals have been recorded as deep as 500 feet. So all that we know about coral reefs is based on the upper 20% of their depth range. And the remainder of their depth range remains largely unexplored. Now, submarines have done a smattering of work in the northwestern Hawaiian Islands, and they could access these deeper reef depths. But chartering a submersible and its support vessel costs something like $40,000 a day. And these subs can go not just to 200 or 300 feet, they can go to 3,000 or 4,000 feet. And generally, if you're going to pay 40 grand a day for a sub, you're going to go as deep as you can, not as shallow as you can. And so subs really don't work in this deeper half of the coral reef habitat. And this is what we call so-called mesophotic coral ecosystems. So we kind of prefer to call it the twilight zone. I realize there's maybe some copyright issues with that, so we don't publish under that name. But in everyday conversation, this really is the twilight zone, because it's deep coral reef habitat, and you're dealing with maybe 1 to 10% of the light that you would get on a shallow reef. So it really is getting kind of into that twilight look where it's getting a little dark. We all carry lights when we do these deep dives. colors are largely absent. But it takes a little more than regular scuba gear to access these depths. So to get us and our exotic dive gear up to the monument, we use NOAA ships. This is the old NOAA ship Hi'i'alakai, which actually is about to be decommissioned in about 2 weeks, but this has been our workhorse ship for almost 20 years for getting up to the monument. So we use, I guess you consider it scuba gear on steroids. These two young ladies are wearing enough gear for 4 normal scuba divers. So when they stand up, they're wearing probably 150 pounds of gear. So it's absolutely awful. It's punishment when you're sitting in the boat with all this weight crushing your shoulders. But as soon as you roll over the side, the gear becomes essentially weightless. It's bulky, definitely. It's work to swim with. It's a lot of drag. It's a lot of windage. But the weight factor kind of goes away and it becomes much more comfortable underwater. We've moved on now from open circuit scuba gear where you exhale your gas away as bubbles to so-called closed circuit rebreathers that recycle the gas that you breathe and inject oxygen as needed. And so they're much more efficient on gas. And the benefit at depth, if you maintain a constant depth, is no bubbles. You don't exhale bubbles. And so it's like skin diving with the sharks. If you're not exhaling bubbles and you're silent, the fish treat you a lot more like another fish. And so the impact of the observer, the impact of the scientist's presence is much smaller on the behavior of the fish. Scuba gear tends to attract some fish that love the bubbles, tends to scare away some fish that don't like the bubbles. And so it has the tendency to skew your data when you're trying to do fish counts. It attracts some fish, repels others, and rebreathers again are silent. So they really are the preferred mechanism for this kind of work. Many of the dives we're doing are at 200 to 300 feet and deeper. And so on shallow dives, you can just put your mask on, look over the side and go, oh yeah, this looks like a nice reef, let's dive here. Not so much when you're in 300 feet of water. You can't just look over the side. You have to go with your sonar or your depth finder on. You drop into 300 feet of water, just go in on blind faith that there actually is a bottom down there. And so there's this kind of awkward, tense moment where you can't see the surface anymore, you can't see the bottom. All you can see are sharks coming up like guided missiles to check you out because they hear you coming and they want to come and see who's knocking on their door. But eventually the bottom comes into focus, and what we get to see down there when we get to 200-300 feet, it's serene, it's calm. You're far deeper than the reach of the swells, so there's no surge. It's largely silent, and it's really, really beautiful. It's almost like a Zen meditation kind of state to get to the bottom in 300 feet. Now, a lot of the structures that we're diving on are old Pleistocene sea levels. So imagine 17,000 years ago during the last great ice age, so much water was tied up in the ice caps that sea levels were up to 100 meters lower than they are now. And so the lower sea levels carved shorelines. And so the structures that we're diving on then are old sea cliffs, very low sea cliffs. When sea levels were 100 meters lower, the ocean carved this shoreline. And then the glaciers melted, flooded these ledges, and now these ledges are 200 to 300 feet. And instead of being shorelines, these are great fish habitats. So these are the kind of structures we're working on. They're very structurally complex, a lot of hidey holes and habitat for fish. And so this is the kind of habitat that we're targeting on these deep dives because that's where you find the structure that will hold fish communities and fish assemblages. Oops. So we get to the bottom. As you can see, this guy has a rebreather on. Really, the diving technology is a talk for another day that Rich Pyle probably should cover. But the rebreather is the thing on his back that you can't see too well. All of these other bottles that he's wearing, all of these other scuba tanks are what we call bailout bottles. And those are tanks that you hope you'll never use. They're bailout bottles because if your rebreather ever stops working properly, if it just plain stops working, or if it gives you a bad gas mix with too much or too little oxygen, you go to these open circuit tanks. So you have to carry enough redundant scuba gear. So if your rebreather failed, you could still get to the surface. And it's a long trip back to the surface because working at these depths, you can't go straight up to the surface and get in the boat. You've absorbed a lot of dissolved gas into your bloodstream. If you go up too fast, it'll bubble out of solution. Exactly like when you open a soda, you release that pressure and it bubbles. Well, if you release the pressure on yourself too fast, your blood will bubble. And if those bubbles lodge in your brain or your central nervous system or your joints, That's what causes so-called the bends, and that is really bad news. So if we spent, say, 20 minutes on the bottom at 330 feet, we'd spend 2.5 hours waiting to get back in the boat, very, very slowly, going back up slower and slower and slower and slower. And so our bottom time where we can get work done is very, very limited, and decompression time can be very, very long. And we do fish counts at these depths by laying out a line and counting all the fish within a meter or two on either side. And we found some really interesting things about levels of endemism. Endemic fishes or endemic plants or insects. Endemics are those species that are only found here in Hawaii and nowhere else in the world. Now, when we started diving in the monument 15 years ago, we were only diving shallow. And on this graph, the light blue bars, and the x-axis is going from southeast to northwest. So we're going further north as you move to the right. The light colored bars are the levels of endemism that we found on the shallow reefs of the northwestern Hawaiian Islands. So across the board in Hawaii, about 25% of our species are endemic, marine species. What you can see on this graph, getting up to Pearl and Hermes, Midway, Kiri Atoll, which are the northernmost atolls, we were getting upwards of 50% of the fish community being comprised of Hawaiian endemics. And that was already considered to be a surprisingly high level of endemism. In a marine system, it's really, really unusual to get that kind of endemism. Now, the darker blue bars are the data we collected when we started diving deeper, 150, 200, 300 feet. And as you can see, as we move up the chain from the Hoa up to Midway, Kiri Atoll, Pearl and Hermes, we're getting upwards of 80 to 90% endemism. In other words, of all the fishes on those deep reefs, 80 to 90% of them are of species that are found nowhere else in the world except here in Hawaii. And that's a level of endemism that is off the scale, or so we thought, until we got to Kiri Atoll, which is the farthest north and the farthest west. For comparison, the Big Island, Hawaii Island, is at about 19 degrees north latitude, 19 degrees north of the equator. Kiri Atoll is at about 28 degrees and some change. And so it's actually 50% further north, 50% farther from the equator than Hawaii Island. And so it's noticeably cooler on the surface, and it's a heck of a lot cooler at 300 feet. And we're doing these dives to 300, 330 feet. We're hitting temperatures in the high 50s. And there's absolutely nothing tropical about that, especially not for us tropical divers in our 85-degree water wetsuits. You know, it's bone-chilling cold at depth. But we did these deep dives at the farthest north atoll, the deepest dives we've done it there. And the amazing thing we found is at 300 feet, these fish assemblages at Kiri are composed of 100% endemic species. In other words, every single fish in this picture is a fish that's not found anywhere else except here in Hawaii. And 100% endemism in any marine ecosystem is simply off the scale. It's just ridiculous. So Kiri Atoll at 300 feet has the highest level of endemism from any marine ecosystem known on Earth. Tropical, temperate, Arctic, deep sea, pelagic, no marine ecosystem on Earth has this kind of high level of endemism. So within the Hawaiian archipelago, which is already thought to be a hotspot for endemism, so many unique species here that are not found anywhere else on Earth. Within that hotspot of endemism, the deep reefs at Kiri Atoll are arguably the hottest part of that hotspot, with just this phenomenal off-the-scale level of endemism. A lot of these fishes, I mentioned that the temperature gets like bone-chilling cold. That facilitates shallower movement by some of these fishes, or so we think. It's a hard thing to experiment with. You can't really change the temperature of the ocean and see what fishes do. But a lot of these fishes, like this Hawaiian pigfish, it's a close relative of the a'awa, or the shallow water Hawaiian hogfish. You know, these have been caught by bottom fishermen, seen from submersibles at 500 to 1,000 feet. And at Kiri Atoll, we're seeing them at only 300 feet, which is still a pretty deep dive. But for these fishes, they've never been seen by scuba divers before, because normally they're far beyond where scuba divers can go. But they're kind of meeting us halfway because it gets cooler. So they come up a little deeper, or rather a little shallower. And we're diving a lot deeper than most folks ever have up in these waters. And so we're seeing some of these really unusual deeper water fish coming up to diveable depths. Same for this little damselfish. This is Strucehaker's damselfish. It originally had been collected by trawls, by dragging the bottom with nets. It had been seen from submersibles at 800 or 900 feet. It had never been seen alive by divers until we started doing these deep dives up at Midway Atoll, Kiri Atoll, at the northern end of the chain. And again, I think really it's because this cold water enables these deeper water fish to come up shallower than you'd find them, say, off of Waikiki. Now, not everything that we come across is known, and that's not surprising because we're going to a place in the northwestern Hawaiian Islands where almost no one gets to go, and we're diving deeper than most people get to dive. And so not surprisingly, some of the things we bring up are actually completely new to science. So this is Rich Pyle. He's actually got a little butterfly fish in his net there. This is at Pearl and Hermes Atoll at about 200 feet. And that little butterfly fish was a new species. And well, the original type specimen came from Pearl and Hermes. This is again a good example of this so-called tropical release, where in cooler waters, the deep water fishes come up shallower, because Rich had actually first observed this fish over 20 years ago from submersibles and submersible video. Now subs are great, you know, you can stay in them, stay dry, go deep, but they're absolutely useless for catching fish. You can look out the window and wave at them or take pictures of them, but until Rich had a specimen in hand, all we had were pictures of this fish, and we finally were able to get specimens at Pearl and Hermes Atoll. And the Bishop Museum actually, I believe, has 3 of these on live display, 3 live specimens, and they've had them for years. And our NOAA facility over in Hilo, the Moku Papapa Discovery Center, also has a couple of these on public display. And the really unusual thing here is that it's really rare to have a fish on public display before it even has a scientific name. So these have been on display for a couple of years before it was even named. But again, this just underscores the fact that we're going to a remote part of the world where almost no one goes, and we're going to depths that very few people dive to. And so not surprisingly, then we're coming back with things that are completely new to science. This is yet another wrasse that is very, very high probability that it's a new species. Before we can ascertain that, we need to collect some material of samples, basically, of its closest relatives, which may be from southern Japan. And well, therein lies the value of these museum collections like the Bishop Museum. This one is not yet in any museum jar, so we'd have to go catch some. But if you catch something that you think is an undescribed species, you can go to museum specimen collections and compare it to every known species of that genus or every similar species of that genus and determine, oh, well, this is actually the same as something that so-and-so described 50 years ago, or confirm that this is in fact something that's completely unique. And the Bishop Museum has one of the most fabulous fish collections. It has far and away the greatest fish collection of Indo-Pacific tropical coral reef fishes in the world. This is another one that we found in 2016. This is Rich Pyle and Brian Green were diving at Pearl and Hermes, and I believe also at Kiri Atoll. And they collected specimens of this. It was a new species of Tosinodes, a small Anthian basslet. The male is the top one, the female is the bottom one. And about the time this fish was first found, this is when President Obama was mulling over enlarging the monument. Remember I said he expanded it from 140 to 580,000 square miles. And so as we were kicking this around and, well, what shall we name this fish? It was like, you know, well, duh, well, let's name it after Obama because he's about to expand the monument. Yeah, that worked great for Rich and Brian because they don't work for NOAA. You see my logo. I work for the federal government, and the policy wonks in NOAA were absolutely not amused that we were thinking of naming this fish after Obama. They were like, my God, you can't name a fish after a sitting president. And I tried to play dumb, you know, like, oh, really? Well, why not? And then, well, that's lobbying. You're trying to influence policy. You're trying to encourage him to enlarge the monument. And I'm like, you know, yeah, of course. Are we that transparent? But it all worked out well because it takes a while to get these things written up. It takes even longer when you send it to a scientific journal for review. They send it out to anonymous reviewers. That's the part that's really beyond your control is how long it'll take the reviewers to review it, what kind of comments and revisions they're going to come back and ask for. So as it turns out, we didn't get this published until, we didn't get it accepted until after Obama had, in fact, enlarge the monument. So I was safe there. It was not an attempt to influence policy because that policy decision had already been made. And instead it was to commemorate this visionary act of enlarging the monument. Also, the timing was good. I think we got it out in December. This was in 2016. It probably would not have been a real good career move on my part to name something after Obama after the next administration was seated. So the timing was perfect. We threaded the window between expansion, the election, and the inauguration. And so now this bears President Obama's name, again, recognizing his visionary act of quadrupling the size of the monument. And it's way above my pay grade to talk to the president, but it's not above her pay grade. This is Dr. Sylvia Earle, the famous oceanographer. When President Obama came out to Hawaii in 2016, he actually took Air Force One up to Midway, took Sylvia Earle with him. And so she presented him a picture of the fish named after him, standing on the beach at Midway, which is kind of cool. Fishes in Hawaii are pretty well worked over. There's a lot of us that are kind of fish nerds. There's a lot of people that like fish. So there actually aren't that many, amazing new discoveries to be made in the fish arena. Whereas for the other 28 or 30 phyla, the invertebrates, relatively few people work on invertebrate taxonomy. In fact, that's a big bottleneck. That's a problem for not just marine science, but science in general, is that we need more taxonomists to identify these things. So the best we can say right now is this sea urchin that we collected is very likely a new species. So it's a close relative of our shallow water vana, the black diadema, the black sea urchins that we see in shallow water, but it's not black, it's not from shallow water, it's from 200 or 300 feet, and it's purple. And so it's probably a new species. This is probably a new species of coral. It's going to take genetics to tease this one apart. Again, we need taxonomists. They're really the experts in this kind of thing are what we need. But the real gold mine for us in terms of new discoveries has not been in fishes or corals. It's been in algae. When you say algae, most people think of green pond scum or something you pull out of the alawai that smells kind of bad. And these are not that. These algae from the deep reefs of the monument, these are some of the most beautiful plants in the ocean. They're colors you couldn't imagine, bright reds and iridescent purples. I mean, these are fabulously beautiful plants. And virtually all of the things we bring up are completely new to science. They've never been seen before. This is a new species. This is a new species. What you can't get from the pictures, this one's very three-dimensional, like a little bush, but it has the consistency of silicone rubber. Like you can squeeze it, bend it, and you can't break it. It's almost like it's made out of rubber. This is a new species. I am a fish biologist. I'm not a phycologist. I just like to collect bright, shiny objects. We turned these all over to Dr. Heather Spalding and Dr. Allison Sherwood, University of Hawaii and College of Charleston, who are in fact card-carrying phycologists who specialize in deep water, deep coral reef algae. And so they help us to identify and name these. This one is actually not officially out yet. It's still in review in the journal. We're giving it the name Laulama ula. So for many of these new species of algae and other taxa, we're actually giving them Hawaiian names through a Hawaiian process as a part of this naming process. So rather than just naming it Hawaiiensis, which has been done quite a bit, that's not an inappropriate name, but it's not too original either. The monument, we have a Native Hawaiian cultural working group, and it's composed of kupuna and community members, and really anyone with an interest in Hawaiian culture and the Northwestern Hawaiian Islands. And they have a nomenclature subcommittee. So we have been going to them to ask for assistance in giving these Hawaiian names. So the formal Latin or scientific name of these will be a Hawaiian name. So for this one, we're kind of duplicating a process that we've borrowed from the Kumulipo. As most of you know, the Kumulipo is the Hawaiian Book of Genesis. It's our creation chant. And many of the species in the Kumulipo are introduced in pairs, where there's a marine and a terrestrial counterpart that share a name, and they also share some characteristics. So this is the fishy example that I like to use. Nohu, the scorpion fish, is the venomous scorpion fish. So it's kind of a prickly fellow because it's got this venomous dorsal spine that will inject you with a very painful toxin. And Nohu, the coastal plant, has these seed pods that are also rather prickly. These will poke you in the foot like kiawe thorns. So here we have the terrestrial and marine counterparts that share this prickly characteristic, and they also share a name. So we kind of borrowed from this process for Saramenia laulamaula. Lau literally translates to leaf. Lama is the endemic native ebony tree, and ula simply means red. So laulamaula refers to the red leaf of the lama tree. And you can see they have some similarities here. The young leaves of the lama tree are kind of oval, paddle-shaped and red, very much like this new species of algae. They also share some similarities in their ecology, in that they're both kind of extremes. Like the lama, the ebony tree is found at oftentimes 3,000 to 5,000 foot elevation. So by Hawaiian standards, that's a pretty high elevation. And conversely, going the other direction, the laulamaula algae is found at 200 to 300 feet. So it's very deep. So they're both kind of from extremes of their habitats. And so again, here we have two species, a terrestrial and a marine counterparts, that share some physical and ecological characteristics and now also share a name. We're going back to the Hawaiian plants here. This is kind of a personal aside, but Rich and I have been lucky to travel to many spots in the insular tropical Pacific. And it's not uncommon to hear the same story wherever you go, which is that, the university scientists came, they got their specimens, they got their data, they went back to America, and we never heard from them again. And that, it's fundamentally is not okay, especially when you're so reliant on indigenous knowledge, on traditional and local knowledge to help you find appropriate reefs to dive on, to help you find these things. And so by going down this Hawaiian naming process for new species, other scientists now are picking up on this, and from taxa as diverse as terrestrial moths and even bacteria from Kaneohe Bay, we now have scientists coming to us going, hey, could you help us come up with a place-based, culture-based name for my new species? And so I think this could fundamentally change the way that science and scientists deal with specimens and data that come from places where there is an extant indigenous culture. Kind of recognizing that these species, we collect them for science, but really in spirit, they belong to the place, they belong to that culture. And by giving it a name that links it forever in perpetuity to that place and to that culture, that gives it a kind of place-based accountability. And again, I think this might ultimately be one of our most significant contributions to science. It's not the new species themselves, but the process of linking them to the culture of that place. Now, not everything we find necessarily gets the Hawaiian name in that way. Rich has named one of his new species after his wife. This one is really personally one of my favorites. Dr. Isabella Abbott, this is especially appropriate for a Bishop Museum audience, who was on the board of directors of the Bishop Museum and as a research associate also on staff. Yeah, Dr. Abbott had the most humble origins. She was born in Hana, Maui, and she went to the very top of her field internationally. And literally, she was the top phycologist, person that studies algae on the planet. She spent most of her professional career at Stanford University. And again, that is absolutely a top tier university, globally speaking. And she was intimately involved in the formation of the Northwestern Hawaiian Islands Coral Reef Ecosystem Reserve. She spent 10 years or more on our Reserve Advisory Council. So I think it's really fitting that an alga from the Northwestern Hawaiian Islands now bears her name. On a more personal note, Dr. Abbott was also one of my mentors. Long story made short, my undergraduate transcript was a train wreck, to put it nicely. And I got turned down the first time I applied to grad school. I was ready to give up. And Dr. Abbott gave me the schooling of my life. And she basically single-handedly got me admitted to grad school. So in a sense, I've been waiting half a lifetime to have this opportunity to be one of the junior authors on a paper that names a deep reef alga from the Northwestern Hawaiian Islands, to name it after Dr. Abbott. And again, there are really tight ties here between Dr. Abbott and the monument, and also between Dr. Abbott and the Bishop Museum. So she really is one of that greatest generation, one of the true giants of marine biology. And now we got a species from the monument named after her. So this whole naming thing, you can have a lot of fun with it. You can go down the cultural path, or you can name it after people that have been significant to our monument, or in this case, to me personally. So I'm going to shift gears now. Most of the picture I've been painting of the monument, as you can see, the big fishes, the big sharks, the turtles, the seals, kind of are representations of the monument as a relatively pristine remote ecosystem that hasn't really suffered at human hands the way some of our reefs here have. And yet we're remote, we're pristine, but we're not immune to global threats that originate outside of our borders and are impossible to manage locally. And that includes, one of the manifestations of climate change, of course, is warmer seawater and big storms fueled by the hot ocean. And this was 2 years ago now, in 2018. Hurricane Walaka passed south of our inhabited island, so it didn't really make the news here. If it doesn't come near Honolulu, then from a news perspective, it didn't exist, right? But at one point in time, Walaka was a Category 5 hurricane. In the past few years, we've had increasing, amazing numbers of very, very large storms, categories 3, 4, 5, enormous hurricanes. So this at its peak was a Cat 5 hurricane that went right next to Lalo, or French frigate shoals. It was almost 9 months before anyone was able to get back to Lalo, to French frigate shoals, to get in the water. And unfortunately, this is what we found. The reef on the left is at French frigate shoals at about 60 feet. And the reef on the right, the picture on the right, was taken within about 5 feet of the picture on the left. So it went from this beautiful 90% coral covered, thousands and thousands of fishes, beautiful coral reef. Really, this is one of maybe the most beautiful coral reef I've ever dived on in the Hawaiian archipelago. And it went from that to this that you see on the right, to just rubble. And the only reason we actually know we were in exactly the same spot is because we had a piece of hardware, a shark tag receiver anchored on the bottom. And so we know definitely we were in exactly the same spot. If it weren't for that, we probably would think, well, we must have missed our spot because this cannot possibly be the same spot. But this is what Hurricane Walaka, this is what a Cat 3, 4, 5 hurricane can do to a coral reef. And again, this really is one of the manifestations of climate change. This is East Island at Lalo at French frigate shoals. This beach used to host 60% of the nesting green sea turtles in the Hawaiian archipelago. And again, I mentioned 90% of the turtles in the archipelago go to French frigate shoals to nest. 60% of those nested on this island. And this was just before Hurricane Walaka. This picture was just after Hurricane Walaka. So a big chunk of sea turtle nesting habitat just went away overnight. Now, we were sort of expecting that with gradual sea level rise, maybe in our lifetimes, but probably over the next 100 years, we would start losing some of these islands to erosion. We weren't expecting it to happen 2 years ago, basically overnight, to be losing islands to climate change and sea level rise. So it's happening. Climate skeptics, really, climate change is real, and it's happening now, and it's happening in our monument. Fortunately, we've been following this with satellite imagery, and over the past 2 years, maybe half of this island has come back. Now, it was heavily vegetated, which really helps to keep the sand in place. So it'll maybe be 10, 20 years before it actually is a fully vegetated, stable island once again. So it's going to be the trade-off between reaccretion of these islands and how fast they can rebuild, and increasing frequency and severity of big storms. And if the storms keep coming more frequently than these islands can recover, then not so much these turtles are going to have some real problems. And then we went further up the chain. We found all of this on our cruise last summer. And when we get to Manawai, the Pearl and Hermes Atoll, this is what we found. An invasive algae, basically overgrowing all of the native algae and coral. Now, I have to be careful when I use that word invasive, because technically I can't use it for this. Invasive, if you say invasive, it means not only is it displacing native species, but also that it's non-native. It's not native to this ecosystem. So we found this algae basically growing berserk and overgrowing everything. The middle of this picture where you see dead coral is where we pulled back some of these algal mats, and everything under it was dead. And so these were in mats that were up to 10,000 square meters each, growing over everything, smothering everything. And when we got back to the lab, we could not identify it. And again, this is Dr. Allison Sherwood at the UH Manoa. Through traditional taxonomy, morphology, and also through genetics, this does not correspond to any known species of algae. So it was described as a new species. And yet, again, we don't even know if it's native to Hawaii or whether it's an accidental introduction from out of the state, but it doesn't correspond to any known species of algae, whether it's from Hawaii or elsewhere. So while we were at Pearl and Hermes, with this really horrific outbreak of invasive algae, we figured, well, let's run some experiments, because we certainly, wouldn't want to bring live algae like this back to Honolulu to do experiments, because geez, if this thing got out, we wanted to make sure that we ourselves as researchers do not inadvertently transport this back to Honolulu Harbor or Pearl Harbor. And so while we were at Pearl and Hermes, where this is already abundant, we conducted a series of experiments, really simple experiments with different dilutions of bleach for varying periods of time. And what we came up with is a 6% dilution of regular household bleach for 10 minutes affected a 100% kill rate. And so again, to prevent inadvertently bringing this back to Oahu, all our dive gear went into the 6% dilution of bleach for 10 minutes. And that, when you scale up to soak all of your dive gear for 15 scientists, is a horrific amount of bleach. And that 6% dilution is still strong enough that it takes the skin off of your fingers. You can feel your hands getting slippery, and that's because it's dissolving your skin. So all of our dive gear, all of our wetsuits, our $10,000 breathers, everything went into 6% Clorox for 10 minutes, just to ensure that none of this hitchhikes back on our dive gear to Oahu. Because I absolutely refuse to go down in history as that guy, the guy that brought this stuff from Pearl and Hermes to Waikiki. It would be an ecological disaster, obviously, but also in Hawaii, it would be an economic disaster if you think about what this could do to tourism. Tourists come to Hawaii to see clean, pristine beaches, to go diving, to go surfing. They don't want to see this. So if this came back with us, it would be an ecological and economic disaster. So a lot of research remains to be done. Where is this algae from? Is it native? Is it introduced? Is there anything we can do to control it? Might it be diminished by herbivores? So there are many years of research yet to be done on this algae, so we're just starting out. So that's pretty much all I got for you today. I don't want to end on that downer note and have everybody kind of think algae is this evil thing that's taking over the world. It's not, just when it goes invasive. So this is my last slide. And again, these are some of the algae we collected on last year's cruise. All of these are new species. All of these are completely new to science. We have many more that didn't even fit on this slide. And again, many of these, if not most of these, are going to go through this Hawaiian naming process. And so their scientific names in perpetuity will be names that are really link it to our place and our culture. And that's all I got for you today. I really appreciate you all spending Friday afternoon with me. I'd be happy to answer any questions if you got them. Otherwise, I'm ready to get me one of those martinis.
Excellent. Thanks so much, Randy. That was awesome. It was a really great talk, and we very much appreciate you sharing your experiences and perspectives on those great trips with you. I really appreciate your shout out emphasizing the need for additional invertebrate taxonomists, because I wholeheartedly agree that this is a major impediment limiting our knowledge of sort of broader marine ecosystems as well as terrestrial ecosystems. So I really appreciate you acknowledging that. And I really like the push to incorporate the indigenous knowledge and honor that through incorporating it into the scientific naming process. That's something we've struggled with ourselves to try to get done. And I'd love to maybe later to talk to you about how we can expedite that process and
yeah, absolutely,
get it done sort of within the timeframes, because as you know, naming species already takes an enormous amount of time. So expediting it to be able to more effectively integrate that would be really nice.
Well, I think it's gaining some momentum, because it started out with me going to scientists who are my friends and colleagues and going, hey, you got that new deep coral from 3,000 feet. Hey, this is Dr. Les Watling at the university. Hey, Les, would you even consider, giving that a Hawaiian name through a Hawaiian process? And he was like, oh yeah, sure. So then I could go to Dr. Heather Spalding and go, hey, Heather, you know what? Look at what Les is doing. Would you consider maybe one of those new species? And it's snowballing to the point where now we have scientists coming to us saying, can you help us go down this path? And so, yeah, I'm really really very, very pleased. And I think this might be one of our biggest contributions to science, really. Is, again, not the new species that we found per se, but this process of linking the new species to a place and a culture.
Yeah, that's awesome. So there are a couple questions. I think Tim asked, let's see if I missed it already. Oh, what are the red spots on the red algae? So I'm not sure what that refers to.
Excellent question. Those are the reproductive parts of the algae where the tetraspores come from. And so a lot of these algae, that is really the definitive taxonomic character that identifies that algae. And so you find a lot of them that have no reproductive structure because they spawned already, or it's not the right season or whatever. And so when you catch one in the act of reproducing, that is the real prize for algal taxonomists, because that definitively identifies that algae through morphology. And we also use genetics. So even in the absence of those reproductive parts, we can still tell if something is the same as something else or something different through genetics. But the complete description, morphologically, it's kind of incomplete to describe a new species without that, which is really the most distinctive character. And again, all of these, just to put in a plug for the museum, all of these will wind up in the museum herbarium. And that really is the value of places like the Bishop Museum to scientists. Is the Bishop Museum is probably the greatest single repository for everything Pacific. There is no place like it on Earth. Rich and I have been to the Smithsonian, we've been to the British Museum. They have great fish collections, but for Indo-Pacific things, they pale in comparison to what Bishop Museum has.
Awesome. Anybody else have questions? Feel free to either chime in by unmuting, or you can always type in the chat and I'll convey them.
I have a question, Randy. It's Alan Ellison.
Yeah, hi.
You have 80% of the fishes in the Northwest Hawaiian Islands are predators?
So 80% and upwards of the fishes at the Northern Atolls are endemic species. 54% of the biomass are apex predators. And so either statistic is pretty phenomenal. When you consider that 3% of the biomass here in the main Hawaiian Islands are these apex predators. So it's an 18-fold difference in the biomass of ulua and sharks and these big predatory fishes. And yet it's really is one archipelago, it's really one species pool, arguably one big ecosystem. And so again, that kind of tells us probably what our reefs here on Oahu used to look like. In fact, I would argue, going out on a limb here, but I think the reef structure, the reef habitat here in the main Hawaiian Islands actually looks better. It's more three-dimensional, more complex, probably in part because warmer water, coral growth rates are much faster. The Northwestern Hawaiian Islands reefs are relatively devoid of that structure. And so I think we actually have better habitat here. I think our reefs here actually could potentially hold even more biomass than the Northwestern Hawaiian Islands. That is if habitat and cover is a limiting factor.
But in a terrestrial system, the biomass of predators is generally far less than the prey. And it seems that you've got more predators than prey.
In terms of biomass, we absolutely do. But again, that doesn't look at the productivity of different trophic levels. If the prey are getting culled off rapidly by these predators, but if they're also being replaced rapidly, what you would see is a very low standing stock of, say, herbivorous fishes. They're getting eaten, so the productivity of the system is phenomenal, but they're getting culled, so you don't see large numbers. It's kind of analogous to a golf course. If you mow it every day, you don't see too much grass, but there's a lot of grass that's growing. So what we don't see in these systems is the amount of growth, the amount of primary productivity, feeding herbivorous fishes. We don't see that. We just see the low standing stocks of grass on the golf course, and the predators, of course, are the big lawnmowers. We do see a lot of lawnmowers.
Okay, well, thank you.
Randy, Tim Gallagher here. I was curious what the relationship is in nutrients between land and sea. Is there a lot of productivity in the marine environment if the land is doing well and if there's a lot of seabird colonies and things like that? Is there that interaction?
Short answer is we don't know. Longer answer is yes, probably. It's something that's begging for someone to do a stable isotope study, to track where this primary productivity is coming from. We're starting to do it on the marine side because on these On these deep coral reefs, there's a lot of fishes, and yet because we're so deep and light is so limiting, there probably isn't much primary productivity happening on the deep reefs. So where are the nutrients, where's the energy and nutrients coming from that fuels these ecosystems? Are the deep reefs actually fueled by shallow reefs? Because some of these sharks and ulua move between shallow and deep reefs, and they might be vectors for energy and nutrients, feeding on a lot of small fish up shallow, bringing these nutrients down deep. Or perhaps it's an oceanic source of nitrogen and primary productivity. Because on those deep reefs, they're numerically dominated by planktivorous fishes. So they're feeding on plankton, whose ultimate source of nutrients is probably offshore in the open ocean ecosystem. But that's a really good question, because to varying degrees, a lot of these islands have enormous bird qualities. There's something like, Rich, help me out here, but isn't it something like 14 million seabirds in the northwestern Hawaiian Islands? And many of those, again, they feed offshore on small bait fish driven to the surface by tuna. They come back to their home islands, nest, and leave those nutrients all over land and the ocean. And so there are certainly spots like around La Perouse Pinnacle at French Frigate Shoals, which is basically a rock covered in guano. And there's a very, very rich algal community within a couple of 100 meters of that rock. And so anecdotally, I think absolutely there's a tight link between seabirds and nutrients and algae and coral reef productivity, but I don't know that anyone's actually looked at it.
Interesting. Thank you.
So related to that, just a couple things. One, Barb mentioned that, with the algae collections, the phycology collections, they have some of those now in the museum already, and she's invited folks that want to come see them to get in touch with her, which is very cool. And then also Tony asked about how you measure the biomass.
We measure it indirectly. When we record fish numbers, we also record fish sizes to the nearest centimeter. And then we use the coefficients that you can take from length and cube it up to a volume or a weight. And so it's based on those coefficients from those or very similar species. We multiply it up to estimate biomass, but we're not actually weighing the fish.
Is there any sort of machine learning way to do that through video capture?
Quite a few folks are working on it and quite effectively. So I think right now it's job security for me because it hasn't been perfected. But some folks in the Gulf of Mexico have been working with AI and learning capacity to identify the invasive lionfish, which you may have heard of. It's a Pacific species that now has taken over the Caribbean and the eastern seaboard and eating all the small fishes. But they have algorithms now that can identify lionfish 2 species and estimated size and weight. And so, yeah, maybe in the next 5 to 10 years, I would think we could be doing video transects rather than with pen and paper, probably getting more accurate results in terms of numbers and sizes and biomass.
That's very cool long term. Chris had a related question on that.
Yeah, I was just getting ready to say that. So Chris asked about using stereo video surveys for estimating biomass and in relation to like Operation Wallacea, incorporating that into their transects.
Yeah, short answer is no, we haven't tried it yet. I know it's more than theoretically possible and people have done it. We've done bait stations that have stereo cameras and have used that to estimate sizes and then biomass. But on diver transects where you're actually swimming with this gear, to have all of this rebreather gear and bailout bottles and 2 digital SLRs and try to even swim with that, it's quite a challenge. So we've been mostly going pencil and paper. It's very low tech, but it's very also effective and cost effective. But we are kind of moving in that direction. We're now doing a 3D photogrammetry for our benthic surveys to characterize the 3-dimensional complexity of the bottom and see how that drives fish assemblage structure. So we're moving in that direction. Corals, rocks, and algae are a little bit easier than fish because they don't move. But the next step definitely is to start applying that kind of technology to fishes.
I should mention real quickly that Ken Longnecker at Bishop Museum has been using this stereo video techniques to try to get estimate fish sizes on transects as well, although I'm not sure how far he's gotten into that.
It's a cool way to do outreach as well, because like the key aspects of it, like the actual making of the video transect with the stereo cameras is easy. It's the actual analysis of that data that is actually the key part, because you have to identify each individual fish that's there to species level and identify the actual individual, and then identify and then sort of work out biomass from that. So it has a lot of potential to be an outsourced community citizen scientist thing, upload the data and get people to watch through it and identify the fish. So it's actually quite a cool thing that may be an idea for a future project.
Yeah, I think so. I mean, it's happening, and I think it's actually really going to come to fruition a lot sooner than I actually realize right now. So.
All right. Any other questions or discussion points? No? All right. So I really appreciate it again, Randy. Thanks for taking the time and sharing that really interesting stuff with us.